Today we're actually going to start working with one of my favorite tools in science, especially in working with genetics, and that is a Punnett square. Punnett squares are a little tool that scientists can use to help them figure out what the possibilities are for the offspring if you know the genotypes for the mom and the dad. And you can actually also work it backwards. If you know what the genotypes of the babies are, that can help you figure out what mom and dad could have possibly been as far as their genotypes. Okay, so a Punnett square, they are used to predict the possible outcomes for offspring when two organisms mix. Now, each organism has a distinct genotype. Remember, that's either homozygous dominant, homozygous recessive, or heterozygous. So they have a distinct genotype for each trait, and you can use those genotypes to see how the babies can inherit the alleles. For example, let's say you have a homozygous tall plant that's dominant, so you're going to be a big T and a big T, is mated with a homozygous short plant. Short is recessive, so that's going to be a little t, little t. And you will have your parents look like this: homozygous tall, dominant, homozygous short, recessive. And if you have that, this is what you're going to get. Okay, now let's look at how this works. So your parents go at the top. Here's one parent, our big T and our big T, and notice we split them. And the other parent goes on the side, little t, little t. Notice they're split. Okay, now what happens is the letter at the top drops. That's where we are with this green here. Okay, see this drop? So this T came to right here, and then it also came down to right here. And the big T here came down here, and it came down here. So the top drops. That's what those lines were for. Okay, now the side slides. And so we're going to take these little T's over here. Here's our two little T's from our parent that was homozygous recessive. And that little T comes over to there. And it comes over to there. And this little T comes over and comes over. So side slides. So whatever is at the top drops into every box underneath it. Whatever's at the side slides across into every box beside it. And so when you finally finish, what you have are the possible genotypes of the babies. In this instant, every single baby would be heterozygous because all this parent can give it is a big T, and all this parent can give it is a little t. Right? And so that makes every baby heterozygous. All right, so let's look at that again real quick in another example. Let's say that both of your parents were heterozygous. Okay? So I have two heterozygous parents. I'm going to draw my Punnett square. I'm going to take one of my parents and split their alleles and put those in the top. I'm going to take my other parent and split its alleles and put it on the side. Now remember, top drops, side slides. So this T is going to come down and come down. This little T is going to come down and come down. All right? So that's going to look like is a big T and a big T, a little t, and a little t, because those have dropped. Now, my side is going to slide. So this big T's got to come across and all the way over here. And this little T's got to come across and all the way over here. All right, so big T comes across. Here's another big T. And here's a big T where it came across. And the little T comes across. And the little T comes across. All right, and it doesn't matter which letter you put in front. What you got to remember is that if you have a big letter, it's going to be dominant. And to have a recessive phenotype, you got to have two little letters. And so what you end up with is heterozygous, two of them, homozygous dominant, and homozygous recessive. All right, top drops, side slides. Hope that helps you figure out how to work them. Ask me questions if you need to.